Okay, so good evening, everyone. So I welcome you all to the week 7 tutorial session of the molecular biology course. So I will wait for 4 to 5 minutes so that learners can join, and then I'll start today's session. So I hope all of you have gone through the week 7 lectures. So in week 7, the topic that was mainly covered was translation. So we learned about translation in eukaryotes and prokaryotes. Yes, so in week 7, we learned about the translational machinery that is present in prokaryotes and eukaryotes. So, what are the various steps in during translation? Like that is initiation. How does the initiation step differ between eukaryotes and prokaryotes? Then elongation and then termination. And towards the end, we learned about the post-translational modification. Right. So let's wait for 2 more minutes and then I'll start today's session. Okay, so let's start today's session. So I will share my screen. So good evening, everyone. I welcome you all to the week 7 problem solving session of this NPTEL course, Molecular Biology. So I'm Akash Jyoti Pathak. I'm one of the PMRF TAs for this NPTEL course. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So yes, we were discussing in the last session, we learned about the transcriptional machinery in prokaryotes and eukaryotes, and also we saw the operon system, how the gene regulation takes place in prokaryotes. Now, in today's session, what we will learn is we will mainly deal with translational machinery that is present in prokaryotes and eukaryotes, and also we will solve a few sample questions that will help you understand the concepts better. Okay, so let's quickly revise what were the topics covered in week 7. So in week 7, the topic that was covered was mainly translation. So in translation, we looked into the initiation. In the initiation, how does the initiation of translation differs in prokaryotes and eukaryotes? What are the different initiation factors that are involved? How is the ribosome recruited? And uh, where will the ribosome now? So where will the ribosome now bind to the mRNA? So there are ribosome binding sites, and there are specific sequences in prokaryotes called the Shine-Dalgarno sequences, to which the ribosome will bind. So this Shine-Dalgarno sequence is present in the 16S rRNA that is present in the 16S rRNA, which is present in the ribosomal subunit, right? So we will look at these one by one, and then we move to elongation and then termination. And in the last part of the lecture we also learned about the post-translational modification. So let's start with the first question today. So the first question is amino acid is encoded by a single. So you have to name the amino acid which is encoded by a single codon. So translation, as you all know that in translation, what is happening? The mRNA that has been synthesized that will be now this is the sequence which is present in the mRNA. So if this is your mRNA, now this sequence which is present in the mRNA that has to be encoded, and the protein has to be synthesized, right? So the genetic information present in the mRNA has to be translated into a protein molecule. So now, what is the? I hope you all know about the concept of codon. So, whenever the genetic code which is present in the mRNA that is read in triplicates, right? So, three uh, uh, three nucleotide bases will code for a particular amino acids, right? So, there will be a start codon, AUG. So there will be a start codon, and then there will be subsequent nucleotides. So these will be read in triplicates, and three nitrogenous bases will code for a particular amino acid. Okay, so why three? So this is a basic concept you must have read in class 11th and 12th. So since there are four different nucleotide bases, and there are 20 amino acids. So there are 20 amino acids. So if four different nucleotide bases has to code for 20 different amino acids, then how many nucleotide bases will be required? So, if there are four nucleotide bases, then this four nucleotide bases uh, cannot code for 20 different amino acids. So if there are So now if there are two nucleotides, so for example, if there are two nucleotides which code for which are coding for proteins, then how many different proteins would you get? So if there are two nucleotide bases, so that so in this first space there can be four different nucleotides, and in the second position there can be four different nucleotides. So there will be 16 different proteins that can be generated by combination of the four different nucleotide bases. But there are 20 different amino acids. So for synthesis of 20 different amino acids, there has to be three. The genetic code has to be read in triplicates so that this would give you. So in the first position, there can be four nucleotides, any four nucleotides. In the second position, there can be either A, U, G, or C, and in the third position, there can be again four different. Any one out of the any four nucleotides can be present, and combinations of 64 by different combinations, you can get uh, 64 different codons, right? So in this question, what is asked is you have to name. So obviously, what is so if there are 64 codons which are coding for 20 amino acids, then it would mean that the genetic code is regenerate, right? So there are more number of codons than the number of amino acids that are needed to be synthesized. So that would mean that one amino acid is coded by more than one codon. So that is what is called the genetic code is degenerate. Okay, so so if you look at there are 64 codons, and if you look at this codon table, you will find that there are two amino acids. So there are two amino acids which are coded by a single codon. One is methionine. So methionine is only coded by AUG, and the second one is tryptophan. Okay. So these are the only two amino acids which are coded by a single codon. Okay. So you see, so this is the genetic codon table. So if you see here, that methionine is coded by a single codon EUG, and tryptophan. Also, if you look at tryptophan, you will see that it is coded by this single codon UGG. Okay. While all other amino acids, you will see that there are more than one codons. So phenylalanine, there are two codons, UUC, then leucine. There are six different codons which are coding for the same amino acids. So remember that the amino acid which is encoded by single codon are tryptophan and methionine. Okay. The correct answer is option C. Now, moving to the next question, during the process of translation in eukaryotes, small unit of ribosome is recruited at. So the options are 5' cap, 5' untranslated region, 3' polytail, and start codon. You have to answer that. So in a eukaryotic mRNA will have a 5' cap, right? So the 5' end of mRNA is modified and it has a 7-methyl guanine triphosphate moiety, right? It has a 7-methyl guanine triphosphate moiety, and the 3' is modified by poly-A-tail, which is polyadenylated by the poly-A polymerase. So you have been asked that now from this mRNA, you have to synthesize the protein. So for synthesis of protein, the ribosomes has to be recruited. So what are the main components that you will be requiring for translation? So first you will require the mRNA, then Second, that will be required thing that will be required are the ribosomes. And the third thing that will be required are the tRNA. So the tRNA acts like an adapter protein between the codons and the amino acids that are to be synthesized. So they are acting as a link between the mRNA and the protein. Okay. You have to here what you have to answer is that the ribosomes which will be recruited for the synthesis of the protein, where would it be recruited? So will it be at the 5' cap, or will it be at the 5' untranslated region? So the mRNA, first it has a 5' cap, then there is an untranslated region. So this untranslated region has the mosaic sequence in eukaryotes and the Shine-Dalgarno sequence in prokaryotes. And these acts as the ribosome binding site, or whether it will be initiate or whether it will be recruited at the start codon. So what is the start codon? So the start codon will code for the first amino acid. The codon which codes for the first amino acid is called the start codon, right? Or whether it will be recruited at the 3' poly A tail. So the answer is option A. So what happens is the eukaryotic mRNAs recruit ribosomes using a specific chemical modification for the 5' cap. And the 5' cap, as we had seen in the last lecture also, this 5' cap is methylated as a methylated guanine nucleotide that is joined to the 5' end of the mRNA via an unusual 5' linkage. And this modified 5' cap recruits the ribosome to the mRNA. So if you look at the initiation of translation in prokaryotes and eukaryotes, so it is a little bit different. So the ribosomes in case of eukaryotes, they are recruited at the 5' cap. Once bound to the mRNA, the ribosome moves in a 5' to 3' direction until it encounters the 5' AUG 3' start codon. And this process is called scanning. Okay. So in prokaryotes, these ribosomes are recruited at the.
Sir, I have a question. Can I ask?
Yes, sure.
Yeah. So in case of eukaryotes, we have seen that the after the transcription of
Hmm.
mRNA,
Hmm.
the 5' and 3', the 5' capping and 3' poly A tail it become modified to prevent from the digestion of endonuclease, right?
Yes.
Yes. So how can you just explain just how the 5' capping or the 3' polyethyl prevents the endonuclease from its cutting activity?
Okay. These endonucleases, they will for the active. So these are basically enzymes, right? So for the activity of these enzymes, they will require a 5' free end or 5' free nucleotide, so that it can act on this free nucleotide and chew it up, right? But if this 5' end is modified by these residues by seven, here it is modified by 7-methyl. This guanine residue is methylated at the at the seventh position of the guanine nucleotide is methylated. So this now this endonuclease fails to recognize this region, and as a result, it will not bind it will not bind to this region, and it will thus prevents it from from getting degraded. Okay.
Okay.
Okay. Okay. Now let's move to the next question. So the interaction between the tRNA and mRNA occurs at dash region of the tRNA. So this is a very basic
Anticodon.
question. So this is all of you know that this is the anticodon arm. So basically, if you look at the structure of tRNA, so this tRNA has an anticodon loop, and this anticodon loop recognizes the codon regions of the mRNA, right? So the anticodon loop will have sequences which are complementary to the sequences of the. It will be complementary to the mRNA or the codon of which is present in the mRNA. Okay. So yes, the anticodon loop. So answer yes, option B. So the anticodon loop contains the anticodon, which is a 3 nucleotide long sequence that is responsible for recognizing the codon by base pairing with the mRNA. Okay, so we will look into the structure of tRNA in brief. So if you look at the structure of tRNA, so this is a 2D structure. The 3D structure, this is a clover leaf structure, right? While the 3D structure is L-shape, it's inverted L-shape. The tRNA has an acceptor stem. So this is called the acceptor stem, this region. Why is it called the acceptor stem? Because it is at this region, the my amino acid will be added. So depending on what is the sequence of the anticodon, that particular amino acid will be attached to this 3' end of the tRNA. Okay, so the acceptor stem is the amino acid attachment site, and it is formed by pairing between the 5' and 3' ends of the tRNA molecule. Okay. This entire structure, this is entire thing is called the acceptor arm, and you can see here that it is formed by the base pairing of this 5' and the 3' ends of the tRNA. So this is actually a secondary structure of RNA that has been formed. So the mRNA suite will have a free 5' and 3', and now because of base pairing or complementary regions between different regions, it has formed a particular secondary structure, and this secondary structure results in this formation of this acceptor arm. So this blue colored region that you see is called the 5' arm, and is called the acceptor stem. And the sequence which is present in this end is 5' CCA 3' sequence at the extreme 3' end of the molecule in a single strand region that protrudes from the double stranded stem. So remember this sequence. So this is CCA 3', and this sequence helps in the attachment of an amino acid to its 3' end. Okay. Then there is the pseudo uridine loop. So this pseudo uridine loop, why is it called? Because it has a modified nucleotide base, pseudo uridine. So that is why because of this presence of modified nucleotide pseudo uridine, this loop is called the pseudo uridine loop. And so you can look at the structure of pseudo uridine. It is similar to uracil or uridine, but it has a particular one of the double bonds is converted into a single bond in pseudo uridine because of the characteristic presence of the unusual base, and the modified base is often found within the sequence 5' P. Uh, this is I stands for pseudo uridine, use CG 3'. So this is the particular sequence which is present in this loop, and then there is the D loop. So so yeah, this is the pseudo-uridine loop, this yellow colored structure, and then D loop is called D loop because it has another modified base which is called the dihydrouridine. So this dihydrouridine modified nucleotide gives this loop D loop, and then there is this variable loop. So this is called the variable loop. So in the variable loop is present between the anticodon loop and the pseudo-uridine loop, and as its name implies, its size varies for between 3 and 21 bases. That is why it is called the variable loop. Okay. Okay. So now there are particular enzymes which are required for recruitment of the or for charging of the tRNA. So what is charging of tRNA? So the tRNA. As was discussed in the lectures also. So the tRNA. So
Option
as we
D.
were seeing that yes. If anyone has any doubt.
Sir, option D, this means answer.
Yeah. Okay. Amino acid tRNA synthesis enzyme present in cells are dash specific. So you are telling that it is mRNA specific. Option B. So what are amino acid tRNA synthesis? So there are basically 20 amino acids, 20 different amino acids, right? Now, for each amino acid, there is a particular amino acid tRNA synthesis. Okay. So, so these amino acid tRNA synthesis, what it does is so as we were looking into the structure of tRNA, so the 3', so you need to add an amino acid at the 3' end of the tRNA. So if this is your tRNA molecule, so this is your 3' end, this is your 5, this is the 5' end of the tRNA. Now, depending on what is the sequence in the anticodon loop, that particular amino acid has to be attached to the 3' end of the tRNA. Okay, so now for attaching the particular amino acid to the tRNA molecule, you require an enzyme which is called amino acid tRNA synthesis. Now, so this amino acid tRNA synthesis enzyme present in cells, they are specific for amino acids. Okay, they are specific for different amino acids, not mRNA. So there are actually more number of amino acid tRNA synthesis than the number of amino acids. So these are called, so there are iso-accepting tRNAs basically. So iso-accepting tRNAs will basically have the same amino acid residue, but the anticodon sequence will be different. Okay, those are called the iso-accepting. So answer is option C. So for each of the 20 amino acids, right? For each of the 20 amino acids, they are attached to the appropriate tRNA. So depending, that is what I was explaining, that depending on what is the anticodon sequence, what is the sequence of sequence of nucleotides in the anticodon loop, that specific amino acid has to be attached to the 3' end of the tRNA. So for attachment of this amino acid, you require amino acid tRNA synthesis. So these amino acid tRNA synthesis are specific for the specific amino acid. Okay. So this is basically the reaction which takes place. So there are the amino acids, so there is the tRNA and amino acid, and for attaching, yes.
I would like to clear my doubt. You have already told this, but still for just confirming, what you said that the amino acid tRNA synthesis, the enzyme which is
Hmm.
uh, taking the suppose glutamic acid to a tRNA and
Yes.
binding with it,
Hmm.
and a enzyme B uh,
Hmm.
taking aspartic acid to another tRNA. So you are
Yes.
saying that both the two amino acid tRNA synthesis enzymes are different, not the same.
Yeah, they are different.
Okay,
They are
so
different. They are different for different tRNA molecules. So so there are around 50 different amino acid tRNA synthesis. Okay.
okay,
So yes. So if it is glutamic acid, then there will be a different amino acid tRNA synthesis. If it is if you have to attach, for example, a serine amino acid, then the amino acid tRNA synthesis will be different. Okay. So so this is the mechanism which takes place. So you have the amino acid, right? So this is the amino acid. So there is an amino group, there is a carboxyl group, a proton, and an R group, right? This is the amino acid. Now this amino acid has to be attached to your tRNA molecule. So here, this is your tRNA molecule. So what happens is, in the first step, what happens? This amino acid is undergoes a process. So it reacts with ATP. It reacts with ATP, and you get a, so this is called adenylation of amino acid. So here what happens, this alpha, so this is your alpha phosphate, beta phosphate, and gamma phosphate. So this amino acid attacks at the alpha phosphate and the beta gamma the diphosphate. This is broken. So you get a, so for in the first step, what happens? The amino acid is attached to an AMP molecule. So this is your AMP, right? Because the two phosphate molecules have been broken. So PPI, so the beta and gamma phosphate has been broken, and this amino acid attacks the alpha phosphate group, and this structure that is formed, that is called the this is what is known as the adenylation of amino acid. So this amino acid is attached to a adenosine monophosphate molecule. Now this adenosine monophosphate molecule, now what happens? This pyrophosphate that is there, this alpha, this beta gamma pyrophosphate is broken, and the energy that is released, that is used for attaching the tRNA, that is used for attaching. So this is the CCA 3 prime, and that I was saying that this is the acceptor arm. So it is this acceptor arm, my amino acid will get attached. So this pyrophosphate that is released, this is broken down, and the energy that is released is used for attaching this amino acid to the tRNA molecule, and the AMP molecule is then released. So this is the overall reaction which takes place for attaching an amino acid to its particular tRNA, and this entire process, this is catalyzed by the enzyme amino acid tRNA synthase. Okay. So the amino acid tRNA synthetase is specific for each tRNA. So there are different tRNA molecules depending on what is the anticodon sequence present in the tRNA, or what is the a particular amino acid it is binding. So there will be different tRNAs. So for each tRNA, there are different amino acid tRNA synthetase, and this entire reaction is catalyzed by this enzyme. And this is called once this tRNA molecule is attached to the amino acid, this is called my charged tRNA. So charged tRNA means my tRNA is attached to the amino acid.
So, sir, for each case, ATP is required. Means for each amino acid.
Yes, for each amino acid, ATP will be required, and first this amino acid will form a bond with AMP. So this is adenosine monophosphate. So first this structure will be formed. Okay, and then this AMP is released, and this tRNA molecule is attached to this via this ester bond, okay, CO bond. So yeah, this is the general reaction. Amino acids. So any amino acid plus tRNA plus ATP, and this is acted upon by this enzyme. This enzyme is catalyzing the reaction, and you get amino acid tRNA plus AMP plus PPI. Okay. Just give me a minute. I will add one figure. Comes clear. Okay, so this is one of the figure from the book Watson, Molecular Biology of the Gene. So you can see here said that there are different amino acid tRNA synthetases. So they are having different structures. You see here that the amino acid tRNA synthetase that is specific for glycine, that has a particular structure alpha 2 beta 2. Then the amino acid tRNA synthetase which is specific for alanine, that is different. So for different amino acids, there are different amino acid tRNA synthetases. Okay, remember this. Yes. So now moving to the next question. During the process of translation, the first amino acid of the growing polypeptide chain enters at dash of ribosome.
Yes, right.
So no, you read the question carefully. First amino acid. first amino acid binds at which site? Enters at which sites? First amino
Is
acid
it
does
P
not bind
side?
at the A site.
Is it P side?
Yeah, it is P site. Yes, it is P site. Okay, the first amino acid will bind at the P site, and then the subsequent amino acids will be recruited at the A site. At the A site. Yes. So if you look at the structure of ribosome, so there are three sites, right? E. So this is the together. So this is the large subunit, and this is the small subunit. This is the small subunit 30s. So if it is a bacterial ribosome, it will have a small subunit 30s and a large subunit 50s. So there are three basic sites. This is a rough figure I'm drawing. So this is the E site, the middle, this is the P site, and this is the A site. So the first tRNA, so the tRNA having the, so this is actually called the initiator tRNA. So the initiator tRNA will have formilated methionine in case of prokaryotes, right? So these were discussed in the lectures. So I'm not going into too much detail. Okay, so this initiator tRNA or the initiator tRNA always binds at the P site, and then the subsequent tRNAs will bind to the A site. Okay. Okay, so the translation initiation is the only time an RNA binds to the P site without previously occupying the A site. This event requires a special tRNA known as the initiator tRNA. So my initiator tRNA will have because the first codon or the initiator codon is what? The initiator codon is AUG. So the AUG codes for which amino acid? Methionine. So the tRNA which will be recruited will have methionine it will be attached to the methionine amino acid residue, right? So but this methionine is different from the methionine which will be present in between the, or which will be present in the open reading frame between the start codon and the stop codon. So that is it has a particular there is a modification. The formyl group is added to this methionine, and this is the, so the first tRNA will bind at the P site. Okay. Okay. So this is clear, right? So yes, so let's quickly discuss the translational initiation. So so this is translational initiation in prokaryotes. So you can see here. So in the first step, what happens? The ribosome. My ribosome, prokaryotes have 70s type of ribosome, and this 70s ribosome is composed of a small subunit and a large subunit. The small subunit is 30s, and the large subunit is 50s. Now, as I was explaining, that the ribosome has three sites. One is the A site, P site, and E site. So E stands for exit, P stands for peptidyl, and A stands for acceptor or accepting site. Okay. So in the first step, what happens? These two subunits dissociate. The 50th and the 30th subunits dissociate, and then there are a couple of initiation factors which are recruited at this position. So now you have the tRNA which is charged with formilated methionine. Now it has three. It can attach to either A site, P site, or E site. Now how would it be ensured that my charged tRNA binds at the P site and not at the E site and A site? So for preventing the binding of the initiator tRNA to the other sites, the initiation factor 3. The initiation factor 3, what it does, it goes and binds to the E site. So once it is bound to the E site, it that this site is not free for binding to the tRNA for binding to the initiator tRNA. Now similarly, the A site gets occupied by initiation factor 1. So this initiation factor 1 occupies the A site, so that the only site now that is left for binding to the initiator tRNA is the P site. Okay. Now after the IF3 and IF1, the initiation factor 2 comes into play. So now in the next step, the initiation factor 2 remains bound to GTP, and this initiation factor 2 recruits the initiator tRNA and allows it to bind to the P site. Okay. So remember that the E site and the A site remains occupied by the initiation factors. So my E site remains occupied by initiation factor 3 and the A site remains occupied by initiation factor 1. Okay. So this figure is taken from the book J.D. Watson. So what happens is the IF1 prevents RNAs from binding to the portion of the small subunit that will become part of the A site. Okay. So basically, it prevents tRNA from binding to the portion of the small subunit that will become part of the A site. And what IF1 blocks the binding of charged tRNAs to the A site. So as you can see in this figure, that my A site is getting blocked by the IF1 initiator protein. So this blocks the A site and prevents the binding of the charged tRNA to this site. Okay. Now IF2 is a GTPase. Okay, so remember this, it is a GTPase. So what it means that this protein will be bound to GTP molecule, and the hydrolysis of this GTP provides the energy for binding to this site, to the P site. Okay, so that interacts with three components of the initiation machinery, the small subunit, IF1, and the charged initiator tRNA. By interacting with these components, IF2 facilitates the association of formylated methionine charged tRNA with the small subunit and prevents other charged tRNAs from associating with the small subunit. So so in the first step, what happened? Only the small subunit is present, and in the small subunit, the charged initiator tRNA will bind at the P site. And who is helping in GTP? Okay, now IF3 binds. Now IF3 binds to the small subunit and blocks it from reassociating with a large subunit. IF3 occupies the part of the small subunit that will become the E site. So the E site is occupied by IF3, and it prevents also prevents the initiated tRNA from binding to this site, and it also prevents the large subunit of the ribosome, 50S ribosome, from binding to the from associating with the small subunit. Okay, thus of the three potential tRNA binding sites on the small subunit, only the P site is capable of binding a tRNA in the presence of the initiation factor. So these initiation factors prevent the initiated tRNA from binding to the other sites and facilitate binding of the initiated tRNA to only the P site. Okay. So the first, always remember that the first amino acid or the first charged tRNA will always be recruited at the P site and not the A site. Okay. Now answer this question: for addition of selenocysteine to the growing polypeptide chain during translation, dash is used. So now this is another example. As I was explaining, that for each amino acid you have a particular amino acid synthetase. So for cysteine, you have cysteine tRNA synthetase. Okay. Then for serine, there is serine tRNA synthetase, tyrosine tRNA synthetase. Okay. So now selenocysteine is a modified amino acid, right? It is not the naturally to the 20 amino acids. These are natural amino acids, and the selenocysteine, it is a modified amino acid. This is also present in our body, but this is modified amino acid. And so how how this modified amino acid, or which amino acid synthetase is responsible for recognizing this modified amino acid? So this is actually a fact-based question. So what happens is selenocysteine is so actually the answer is option B, serine tRNA. Okay. So the selenocysteine is not incorporated into proteins by chemical modification after translation. Instead, selenocysteine is generated enzymatically from serine, carried on a special tRNA that is charged by serine tRNA synthetase. So so there is this serine tRNA synthetase. So this serine tRNA synthetase helps in the so this serine tRNA synthesis will recruit serine, right? It will recruit serine, and then this serine is chemically modified into selenosystine. So unlike standard amino acids, the selenosystine is synthesized directly on its tRNA and inserted into proteins in response to specific UGA codon. So this is actually a stop codon, UGA codon, but this stop codon, it also codes for selenosystine, which typically signal translational elimination, termination. Okay.
So, sir, I would like to ask a question that
Mm-hmm.
will this in a natural in vivo system of an
Mm-hmm.
animal cell or whatever, so where UGA is present, so we know that for the stop codons, no tRNAs will come for that to the mRNA. But
Mm-hmm.
will for UGA the selenosystine will come or not?
Okay, so yeah, so if UGA is present in between the codon, right?
Yeah,
That is what you're
yeah.
asking.
And
So,
in the natural
so will
system,
it? Hmm. So will it code for selenosystine, or will the translation
it
stop
depends
at
on
this
the
site? So
method.
how is it decided? Right.
Yeah.
Okay. Uh, this is actually a good question. So there are this mechanism, how it exactly takes place. I have to read about it. I I also don't know. So I will read about this and I'll let you know. But there is a particular mechanism which takes place, and by this mechanism, this is this translation, or when it has to code for selenosystine and when it has to stop, that is taken care of. Okay. Um, so there are basically, so if there are special tRNAs which are recruited for this. Um, so I will see the so how this is ensured. Yeah,
Okay, sir.
look for the answer. Yeah, actually, yeah, that is something. That is a good question. I have to see what is the mechanism which takes place to ensure that the translation is not stopped or terminated, but selenosystine is incorporated at that position. Okay. Now, so we'll discuss it in the next class. And this question again, I'll discuss. Um, next thing is translation initiation in eukaryotes. So these things are actually um, there are many proteins. You have to remember what is the function of these different initiator proteins or initiation factors. Which initiation factor is involved in recruiting the tRNA, which is involved in recruiting the mRNA. So there is nothing much to understand over here. The initiation factors that are present in prokaryotes are different from that in eukaryotes, but mainly here also, here the initiation factors that are present, they are called the EIF1, EIF2, EIF3. Okay. Similar to what the initiation factors which are present in prokaryotes, IF1, IF2, IF3. Here also you have three initiation factors, EIF1, EIF2, EIF3. Other than, so these are the main, and other than that, there are also other initiation factors, IF4, IF5. Okay, so this initiation, so as I was explaining in prokaryotes, that IF1 and IF3 blocks the E site and the A site. Here also the IF1 and IF3 is blocking the A site and the E site, and EIF2 helps in the recruitment of the initiator tRNA to the E site. Okay. For the initiator tRNA. So in eukaryotic cells, the binding of the initiator tRNA to the small subunit always precedes association with the mRNA. Okay. So here the translational initiation in eukaryotes, it is different from prokaryotes. So the ribosome So the ribosome will not directly associate it will not directly associate with the mRNA in the first step. So the ribosome will first bind to the initiated tRNA, and then there will be certain modifications which will or certain other factors which will be required for recruiting the mRNA to this a small subunit of ribosome. So that is what is written here, that binding of initiated tRNA to the small subunit. So first binding of initiated tRNA to the small subunit takes place, and then the association of mRNA takes place. Okay, but in prokaryotes, first the mRNA. You have the mRNA, and there is a ribosome binding site on the mRNA, and here the ribosome will bind. First, the ribosome will bind, and then the initiated tRNA is recruited in case of prokaryotes. But in case of eukaryotes, the mRNA, the ribosome will not go and directly bind to the mRNA. First, the ribosome. So if this is your A site, A site, P site, and E site. So first, the initiator tRNA will be recruited at the P site, and then there will be other elongation. There will be other initiation factors which will be required for recruiting the mRNA onto the ribosome. Okay, so that is what is written. Second, a separate set of auxiliary factors mediate the recognition of the mRNA. Okay, so there are other factors which will be required for the recognition of mRNA by the ribosome. Okay, third, the small ribosomal subunit bound to the initiator tRNA. Small ribosomal subunit bound to the initiated tRNA scans the mRNA for the first AUG sequence. And finally, the large subunit of the ribosome is recruited after the initiated tRNA base pairs with the start codon. So after the initiated, so the large sub, so after the large subunit of the ribosome is recruited, and then this ribosome will scan the mRNA sequence. And once it finds the AUG sequence, it will then following that the initiator tRNA. Uh, uh, so no, first the initiator tRNA is recruited, and then and once the ribosome gets this AUG code, then it will associate with the large subunit of the ribosome. Okay. So that is what is explained. The third step, what happens? The small ribosomal subunit binds to the initiated tRNA and scans the mRNA for the first AUG sequence. So you have the small subunit of the ribosome. So the small subunit of ribosome is 40S in case of eukaryotes, right? So it is 40S. Now through the 40S small subunit of the ribosome, the initiated tRNA will bind at the P site, and then this ribosome and this initiated tRNA complex, it will scan the mRNA for the AUG sequence. And once this AUG sequence is obtained, the large subunit of ribosome is recruited, and you get a closed complex. Okay. So in the first step, what will happen? Similar to what was happening there, that the uh, so there also that there was dissociation of ribosome. So here the dissociation of ribosome takes place in so the 80th ribosome is dissociated into the 60th and the 40th subunits, and there are four initiation factors: E, I1, EIF. E stands for eukaryotic. Eukaryotic initiation factor 1, eukaryotic initiation factor 1A, eukaryotic initiation factor 3, and eukaryotic initiation factor 5, which will bind to the small subunit. Now, eukaryotic initiation factor 1, 1A, and 5, they act in an analogous manner to the prokaryotic initiation factors IF3 and IF1. So this is, I said this
Sir,
wrong. This is actually IF. Yes.
sir, in the starting of the initiation, the two ribosomes are dissociated, and
Yeah.
then again it will get associated, and then again it will get dissociated.
Yes. So before the starting or the initiation, or before the initiation of the translation process, the ribosome is dissociated, and then during the elongation step, before the elongation step, again it will form the 60S and 40S will form a complex, right? And
No, sir.
after the
In the case of initiation, only first it will get associated and then dissociated, and again it will get associated. Means, sorry,
yes,
first it
yes.
dissociated in the initiation process only.
Yes, in the initiation process only. First, it will get dissociated, then it will get associated. Before, so as soon as the elongation process is about to start, it will get associated. Okay. And then after the termination process, the two subunits are again dissociated. Okay.
Sir, but in the case here, it is talking about initiation or the whole process in
No,
this.
here it is only talking about initiation,
So, sir,
only the
in the
initiation process.
third step, it is written that dissociation of ribosome into 60N and 40S.
Uh, this point. So
Third point.
here, so these are fourth point. Now, so this is the initiation. You can break down the initiation step into three sub steps. Okay. So yeah, you can break down the initiation, the translation initiation of eukaryotes into three steps. So in the first step, what happens? First step, the initiated tRNA. So if this is the small subunit of the ribosome, and
No, sir.
this is your
I am asking that my question is that
fourth step. No. So this
no,
is the
sir.
first. So this is different. These 3 points are different from the fourth point. So the fourth point is again starting. So this will. So this is actually a summary of what happens in the initiation, and then I have discussed what happens one by one. Okay.
Okay, sir.
Yeah. So this 3 points. So the first 3 points I have just added to give a basic understanding of what all happens in during translation initiation, and then I have described each step in detail. Okay. So in the first step of initiation, what will happen? The ribosome, you have the 80S ribosome. The 80S ribosome will dissociate into the 60S ribosome and the 40S ribosome. Okay. So now this 40S ribosome, this is the small subunit, and the 60S ribosome is the large subunit. It is the last subunit. So first, what happens? So this is your small subunit. This is the 40S. So in the 40S at the P site, so this is A site, for example, this is E site. So at the P site, what happens? The initiated tRNA is recruited. Okay. The initiated tRNA is recruited. And then, so for this, which initiation factors are required? So there are these three initiation factors, EIF1, EIF1A, and EIF5. So these three proteins, they act in an analogous manner, similar to what we had seen in prokaryotes. So in prokaryotes, there was initiation factor 1, 2, and 3. You have three initiation factors, which are IF1, IF1A, and IF5. So these are acting in an analogous manner and preventing the large
Sir, I can't hear you.
RNA from binding to the A site. So here also, okay, so here also the same thing is happening. So the first amino tRNA will be recruited at the P site. Okay, so for this so that my first tRNA binds at the P site, this A site and E site remains occupied by these initiator factors. Okay, and the initiator tRNA is escorted to the small subunit by the three subunit GTP binding protein EIF2. Now, EIF2, what it does, it binds the initiated tRNA only in the GTP bound state. So when this EIF2 is bound to GTP, so I will explain this in the next slide. So in the next slide, there is a figure. So once you go through this figure, you will understand. You can understand it better. So when this initiation factor 2 is bound to GTP, only then this initiation factor 2 goes and binds to the initiated tRNA. Okay, and then the association of this EIF2 initiated tRNA and the small subunit of ribosome. So these three structures together these three structures together is called as the ternary complex. So this is what happens. So So you see here, you see this figure. So this is the large subunit and small subunit. So in the first step, what happens? The large subunit and the small subunits has dissociated. So this is your small subunit and this is the large subunit. Now, what happens? You see here that the A site and the E site, the A site and E site remains occupied by the initiation factor 1A and initiation factor 1. So this initiation factor 1A and initiation factor 1, these two initiation factors, they occupy the E site and the A site so that the initiator tRNA can only bind at the P site. Okay. Now here you see this is your initiator tRNA, this structure. So this is your initiator tRNA, which is bound to methionine. Okay. Now this initiator. So what happens now to this initiated tRNA, the EIF2 GTP. So EIF2 when this eukaryotic initiation factor 2 is bound to GTP, only then it will associated with the initiated tRNA. So now EIF2 GTP, it helps in the recruitment of the initiated tRNA to the P site. Okay. P site, and this is all called the 43S pre-initiation complex, or this is also called as the ternary complex. So because why is it called the ternary complex? Because there are three structures here. One is the small subunit of ribosome, one is the initiator tRNA, and another is your EIF2 protein, which is bound to GTP. So together it is forming the ternary complex. And then what happens, as I was mentioning, that here in eukaryotes, the initiation step is different from that in prokaryotes. Why? Because this ribosome is first attached to the initiator tRNA, and following this, the mRNA is recruited. So for the recruitment of mRNA to this ternary complex, you require other initiator proteins. Other initiator proteins, what are these initiator proteins? These are EIF4, 4E, 4G, 4A, 4B. So these different proteins are required, and these proteins together, they will recruit the mRNA to the 43S pre-initiation complex, resulting in the formation of a complex which is known as 48S pre-initiation complex. Okay. Now, once this 48S pre-initiation complex is formed, then my large subunit will come and you will get the 80S ribosomal subunit. Okay. So this is actually, there are many steps involved. We have to go through this again. Um, so you will understand this, what is happening. You have to go on your own. You have to read and understand once again on your own. So all in all, what is happening is, first you have this small subunit and large subunit dissociate. You get the small subunit, and then the small subunit are acted upon by various initiation factors, so that the initiated tRNA now only binds to the P site. Now, once this complex is formed, there are other initiation factors which are required, which are EIF4E, EIF4G, EIF4A. So EIF4E, EIF4E initiator protein recognizes the 5' cap of the mRNA, and then on. So in a separate series of reactions, the mRNA is prepared for recognition by the small subunit. So you have to prepare the here what happens, the mRNA has to be prepared so that it can be recognized by the small subunit of ribosome. So for this, what is required? This process begins with the recognition of the 5' cap. So you remember this protein. So the 5' cap of the mRNA is recognized by EIF4E. EIF4E. So you can see in this figure also that this 5' cap of the mRNA is recognized by this initiation factor EIF4E. Right, and then there is series of additional initiation factors. So what are these? These are EIF4G, then EIF4A. So EIF4G, what it does, it binds to both EIF4E. So this is your EIF4E. It will bind to EIF4E and the mRNA, whereas EIF4A binds EIF4G. So you see here, this is your EIF4A. It is binding to EIF4G and the mRNA. Okay, and then finally there is another initiation factor, which is EIF4B. Okay, so all these factors together help allow the mRNA to be recognized by the small subunit of ribosome. Okay. So now answer this question. So during translation in eukaryotes, binding of initiator tRNA at A site is prevented by the activity of. So remember that it is similar to what we did in prokaryotes. So in prokaryotes, there were three initiation factors, IF1, IF2, and IF3. So these are present in prokaryotes, and in eukaryotes, you have four initiation factors, which is EIF1, EIF1A, okay, EIF3, and EIF4, right? These are the three, EIF3 and EIF5. EIF3, EIF5. So if you see this figure carefully, so you see this carefully and answer that
Sir,
which
EIF
हाँ
4A.
EIF 4A होगा कि 1A होगा? You see here which is the initiation?
1A, sir. 1A
1A.
in A site and 3 in E site.
Yes. One initiation factor 1 is binding to the E site, and initiation factor 1A. 1A is binding to the A site. Okay. So answer is initiation factor 1A. Okay. So initiation factor 1A is blocking your A site. You see, this A site is blocked by initiation factor 1A. Okay, so these are actually things you have to remember. There is nothing much to understand here. So you remember that there are two initiation factors, namely EIF3 and EIF1A. So EIF3 and EIF1A binds to the newly formed 40S subunit, thereby blocks its reassociation with the 60S subunit. There is option C. Okay. So first step is ribosomal dissociation. This is directly from the lecture slides. I have taken this figure. So the 80S ribosome will dissociate to form 40S and 60S subunit, and then following this, the 40S small subunit which is present here at the A site and the E site, there are two initiation factors that will come and bind. So at the A site, the EIF1A will be present, and at the E site, EIF3 will be present, and this blocks it from reassociation with the 60S subunit. Okay. Now 5' cap found in eukaryotic mRNA is recognized by. So look at this figure. So you see here that the 5' cap of the mRNA, 5' cap of the mRNA is recognized by which initiation factor? Which
ELF
initiation
4A.
factor? So this is this one, EIF4E. So EIF4E is labeled as E in this figure. Okay. So there are many initiation factors over here which are involved in different roles, which are having different roles here. So one, there are initiation factors which are recognizing the mRNA and recruiting it to the ribosomal subunit. Then there are initiation factors which are blocking the different sites present in the on the ribosome. Okay, so you see here EIF4E. EIF4E recognizes the 5' cap, and following this EIF4G comes and binds at the EIF40 and the mRNA, and after this the EIF comes, EIF4A and EIF4B. Okay. So yes, the answer is this same figure. So the mRNA is prepared for recognition by the small subunit. So the mRNA is not directly recognized by the small subunit of the ribosome. So you have to prepare the mRNA so that it can be recognized by the ribosome. And for preparing this mRNA, there are a couple of initiation factors which are required. So this process begins with the recognition of the 5' cap by the cap binding protein EIF4E. So EIF4E is the cap binding protein, and then this EIF, following EIF4E, the EIF4G is recruited, which will bind to both EIF4E and mRNA, and after the EIF4E, EIF4A will bind to EIF4G and mRNA, and then EIF4B will come. Okay. Now after this, so first you were getting a 43S initiation complex. So what is the 43S initiation complex? So the 43 initiation complex was a ternary complex, right? It had so this is your small subunit of ribosome. This is what? This is 40S. So the 40S small subunit of ribosome, the initiator tRNA. So for example, this is your initiator tRNA, which is bound to methionine. Okay, which is bound to methionine, and then EIF2 or EIF2. So together, these three proteins will form the initiation, will form the 43S complex. Now, after this 43S initiation complex is formed, once the mRNA comes and binds to this 43S and initiation complex, the structure that is formed, that is called the 48S initiation complex. Okay. So what is the order you have to answer here? So first, the correct sequence of formation of 48S initiation complex. So if you understood this figure, you can answer it. So first, what you are having? You are having the
Option
40S right, option
C.
C or D. देखो सबसे पहले क्या आया? So in the very beginning, you are having the 40S ribosome, है ना? उसके बाद कौन सा आया? उसके बाद tRNA आया, इनिशिएटर tRNA, ठीक है? और उसके बाद लास्ट में कौन आ रहा है? mRNA, है ना? देखो ये फिगर देखो, ये फिगर देखो, इसमें इसमें समझ आ रहा है? सो फर्स्ट यू सी, आ गया ना समझ में?
हाँ सर।
हाँ देखो, पहले ये डिसोसिएट किया, डिसोसिएट करके स्मॉल सब यूनिट और लार्ज सब यूनिट में डिसोसिएट कर गया। उसके बाद ये जो स्मॉल सब यूनिट आया, ये स्मॉल सब यूनिट के पी साइट पे आके इनिशिएटर टीआरएनए बाइंड कर रहा है, ठीक है? और इसके बाद आके तुम्हारा एमआरएनए बन रहा है। रुको, लेट मी ऐड अ फिगर दैट यू कैन अंडरस्टैंड, जस्ट गिव मी अ मिनट। 48, I will add a figure of the 48 initiation complex. Uh, so now see this figure. Actually, this figure I could not adjust it, adjust the entire figure on the slide. So I have cut this figure and I have added here. So actually, what is happening is, so this is your 43S in pre-initiation complex, and this is the mRNA to which these initiation factor EIF4B, EIF4G, these factors are binding, and then this 43S initiation pre-initiation complex and the mRNA together when they are coming, this structure is called the 48S initiation complex. Okay, so the 48S initiation complex means इसमें क्या क्या है? एक स्मॉल सब यूनिट ऑफ राइबोसोम है, एक एमआरएनए है और एक इनिशिएटर टीआरएनए है. So order. So you have been asked the order in which the 48S initiation complex is formed. So the order will be, first you will have the 40S small subunit of ribosome, then the initiator RNA will be recruited, and then the mRNA will be recruited. Okay, so that is the order. So answer is option number. Option A or option D. So both the options are same here. Okay. Did you understand, Samiksha?
Yes sir.
Okay. So have you solved the assignment for week 7?
No sir, not yet.
Okay. So the assignment questions are easier than what I'm discussing. They're comparatively, they're easier.
So actually, it's completely hodgepodge.
It is harder. Okay. Okay. Okay, let's discuss
No,
this.
harder,
So
sir, but
by
mixed hodgepodge. Can't
okay,
remember the series, means sequentially can't remember the sequence
yes,
properly.
actually, yeah. They are difficult. I understand they're even difficult to remember for me because there are so many initiation factors. Some are they are different in prokaryotes and eukaryotes, and you have to remember the names. It is actually difficult to remember
So in
what.
one go, when I saw the video, then it was
Hmm.
completely out of my mind. What to
Yeah.
means, what to learn and what not to. Then again, I watched the video. Then also, it's not getting clear completely.
Hmm. Which part are you facing difficulty?
Means sequentially, I can't remember that this is occurring after this and this is happening before this. Like everything is getting mixed up completely. I think I have to watch
Actually, you
some
can go
other.
through few animation which are
Yes,
present on YouTube. They are on YouTube. There, if you go through the animation, then it becomes easy to remember. So
yes.
once you understand the picture and it is to visualize, it becomes easier to visualize to go through these animations because these are very complex mechanisms which are taking place. So which one is recruited first, and then which second, which protein will be coming, and then
And
which will
after
bind to
that,
mRNA.
there is difference between prokaryotes and eukaryotes.
Yes,
After
the
after that,
names are also different. Uh,
yes.
yes, actually, there is a lot of confusion that happens. So, actually, when you are discussing each things separately, translation, transcription, then these specific questions come. You have to remember these things. But when the entire course is completed, there will be many other things you have to remember. So these minute, detailed questions does not usually come, right? Only things which you have to understand the concept or the topics where understanding the concept is necessary only in those parts the questions are asked. Okay, you don't have to worry. So because in every week we are reading a particular topic, so from that topic, every minute detail or each line from each line questions are framed in your assignment. That is why it becomes difficult.
Actually, I'm not giving the exam, but then also just for my knowledge, I am
Knowledge.
watching the videos and
Yes,
the
yes.
course also. And I was not joining since means your class because in my Gmail the link was not coming, and I was thinking that you are not taking the class only. And then I when I
Okay.
looked at the announcement section, then yesterday I think, then I saw that there is a session from 7 to 9, means I was missing since last 4 or 5 times the session.
Yes, last two lectures there were very few. No one had joined actually. In the last lecture, no one joined.
No, sir, saw the attendance. One
But
has joined, and in the last to
yeah,
last video, one also not joined. I was
yeah.
not getting the link only. I means I
Okay.
should have seen that announcement section, but I was not seeing that announcement section. I was
Usually
only
you receive a reminder mail on Monday.
no, sir. I am not
You
getting
didn't
that
receive
mail.
a reminder mail.
I am
Okay.
getting only the mail from 6 to 8 p.m. one, but 7 to 9
Oh,
I am not getting. Maybe my mail problem or something issue is there.
okay, okay. Okay, no issue. So the parts which you are facing issue, we will I can discuss those parts more um, uh, or break down those parts and discuss it more easier so that it becomes easy for you to understand. Okay.
Okay, sir. So I am thinking that what I will ask to you and what I will not ask, because it is very much complicated. Means it is getting complicated for me. Means
Yes.
slowly it's getting clear little bit, but then also
Hmm.
completely clear. So
Hmm. Yeah, actually, in 2 hours, I cannot complete
And
the
sir,
entire week what
this
has been
session.
taught. So just a few questions I discuss. I cannot
Yes,
discuss go through all the topics which were covered. Right. So that
and
is
sir,
why also it becomes confusing sometimes because
and sir,
just now I
this
was
session.
discussing something and now I'm switched to prokaryotic. So I was discussing prokaryotic initiation, then I started discussing eukaryotic initiation. So that is why it becomes confusing sometimes.
And sir, I am finding that this video is quite speedy. Means I think from the starting only you are speaking so fast that I am unable to catch you. I think it's so in 1.5x speed, so it's getting a
Okay, okay.
little.
I will go slowly. I will go slowly. Okay.
So at least one concept, if I understand, then also if I understand it clearly, then I can be able to answer all the questions from that section only. And from little bit from this and little bit from this and bit from this, then I will not get anything.
Okay,
So
okay.
you just keep on going. The
Okay,
means
I will.
little. Huh.
Okay, actually, the questions that has been given, they are from so different parts. So that is why I feel that I explain one particular part and then I have to go to the other part. So I cannot explain all the concepts which are there in that uh, particular topic.
Okay,
Okay.
okay,
So
sir.
yes,
Okay,
what I will do, I will take care of this. So from next week, I will discuss a particular topic in detail so that particular portion is clear. So you can answer all the questions. If any question is asked from that particular section, that you can easily answer those questions, or the concepts related to that part becomes easy for you. Okay. Actually, these things are
okay.
there are not much concepts you have to understand here. There are a lot of things you have to remember. There is more number of points or names of different proteins you have to remember here than what you have to understand. That is why.
And sir, means sometimes the questions are from the same topic, like this question and the initial and the previous question. This
Hmm.
continuously two or three questions are from the same topic. If
Yes,
earlier I would have understood, then I could answer these questions.
yes, yes.
That's the problem.
Yes, that is why what I do is I first explain the concept and then I ask the question so that once if you have understood the concept, then you can try out the question and see
Yes,
if
that
you have
that is
understood
why
it.
I always see your video leaving others. But today I was thinking that you have started from the questions itself.
Yes, actually, yeah.
So
I
not
thought that
means a little bit also you can explain and then you can move. Just a suggestion that little bit
yes.
also you can explain and then you can move to the questions.
Okay, okay. Yeah, okay, I will take care of that. So this today's presentation, I have added more number of questions rather than discussing the concept. So I will take care from the next class. I will add the concept. I will explain all the concepts and then I will ask the questions related to those concepts. Okay.
Okay, sir. Thank you, sir.
Yeah. So this question you have understood, right? So
Yes,
first you
yes.
will first the 43S initiation complex is formed.
And now this question no need to explain. You just
Okay,
move.
okay,
That.
okay, okay, okay. So the answer is option A and option D both are same actually. Yeah, this question. So it is either A or D. Okay,
Yes, sir. So I got this question.
okay.
Yes, sir.
So you can go through this slide also. Here also it has been explained
Yes,
very
I
nicely
will
in the
go
lecture
to
slide.
the video.
Okay,
Yes, sir.
okay. I'm moving forward then. Okay.
Yes.
Okay. Now, recruitment of 60S ribosome to the 48S initiation complex is facilitated by. So now what you have to understand is you have the ribosome. Okay. You have the ribosome. The ribosome to this ribosome, the T initiated tRNA is attached. Okay. So this is your E side. This is A side. This is E side. Now, following this, what has happened? The mRNA has also been recruited. Okay.
So
The
this
mRNA.
translation I think was EIF 5B.
Yes, it is actually AIF5B. So once you get the 48S initiation complex, the next step is what? Tell me what will be the next step?
48S means from the starting mRNA
No,
plus
no, yeah, this
48.
is your 48S initiation complex, right? This is the mRNA. This is your small subunit of ribosome, and this is your initiated tRNA. The next step will be what? The next step will be you have to recruit the large subunit of ribosome, right?
Mhm.
So that is what. So recruitment of 60S ribosome to the 48S initiation complex. So this is your 40S initiation 40S subunit. So now to this 40S subunit, mRNA, the mRNA is has been recognized by this small subunit of ribosome, and the initiated tRNA is also attached. Now the next step is recruitment of 60S ribosome. So that recruitment of 60S ribosome is done by which initiation factor? It is IF5B. Okay.
B.
Hmm. So now you have this entire complex, 80S complex, right? This 80S ribosomal complex. Now, once this entire complex is found, then the elongation can start. Okay. Yes. So these questions are directly from the slides actually. Okay. So this 48S initiation complex binds to the 60S ribosomal subunit to form the 80S initiation complex. Okay. Now this will involve what? This binding involves the hydrolysis of GTP. Okay, bound to EIF2. So as I was explaining that always remember that IF2. So IF2 in prokaryotes was also bound to GTP, and in eukaryotes also the same EIF2. EIF2 is bound to GTP. Now for the association of the 40S ribosome subunit to the 60S ribosomal subunit, you will require the hydrolysis of the GTP. Okay. This step is facilitated by the involvement of EIF5. Okay.
Means the GTP which was bound to that tRNA will now get hydrolyzed.
Yeah, now it will get hydrolyzed, right? This EIF GTP is involved in recruiting the initiator RNA to the P site. It was only EIF2 है ना, ये सिर्फ इनिशिएटर टीआरएनए को पी साइट में लेके आ रहा है, ठीक है। अगर अगर ये जब तक EIF2 GTP के साथ नहीं बाउंड होगा, तब तक ये इनिशिएटर प्रोटीन नहीं आ पाएगा। मतलब ये EIF2 ही ये इनिशिएटेड टीआरएनए को पकड़ के पी साइट पे लेके आता है। ओके, इज इट क्लियर? Now, but for
Yes.
the association of the 60S ribosome with the 40S ribosome, you require EIF5. Okay.
So initiator means the initiator tRNA was already present earlier, no, on the P site. So why
Yes,
EIF5?
it was present. It was present. So that is what I'm explaining. It was present at the P site, but this GTP will only be hydrolyzed when it has to associate with the 60S subunit of the ribosome. Okay. मतलब
मतलब
जो है
उसको
ना,
उसके संग जुड़ने के लिए जीटीपी
हाँ
का निकलना जरूरी है।
हाँ, ये EIF GTP जो है ना, ये इसको पी साइड पे लेके आ रहा है, और ये GTP इसके इसके साथ बाउंड रहेगा, ठीक है। जब 60S राइबोसोम आके इसके साथ जुड़ेगा, तब ये GTP हाइड्रोलाइज होगा, और तब ये कॉम्प्लेक्स बन जाएगा 80S, ठीक है। एंड वंस दिस GTP इज hydrolyzed, this EIF2 protein initiator factor, this will also get dissociated. Okay. So the only function of this EIF2 GTP is for the recruitment of the tRNA, and this GTP function of this GTP is to that hydrolysis of this GTP will result in the release of energy that will recruit the 60S subunit of ribosome. ठीक है, ये यहाँ पे बाइंड होके रहेगा तब तक जब तक ये 60S राइबोसोम ना आ जा रहा है, ठीक है।
तो EIF5 का काम वही हुआ।
हाँ, वही हुआ। ये EIF2 का बस वही काम है कि वो इनिशिएटेड टीआरएनए को लेके आएगा पी साइट पे।
ओके,
ठीक
एंड
है। और
EIF5
EIF5 का क्या है, वो ये 60S जो लार्ज सब यूनिट है ना राइबोसोम का, उसके साथ एसोसिएशन में हेल्प कर रहा है। ये जो 80S इनिशिएशन कॉम्प्लेक्स जो बनेगा 40S और 60S जोड़ के, पहले डिसोसिएट हुआ, फिर से एसोसिएशन होना पड़ेगा। सो फॉर द रीएसोसिएशन और फॉर द एसोसिएशन ऑफ द 60, हाँ, EIF5 is required. Okay.
तो स्टार्टिंग में जब एसोसिएशन हुआ था तब at that time nothing was required, or at
No,
that time I think
initially only dissociation happened, right? Starting में तो dissociate हो गया 60s or 40s.
हम्म
Now you have to again, so after dissociation, once you have the 48S initiation complex, after the formation of the 48S initiation complex, now you have to form the 80S initiation complex. So for formation of, for reassociation of the 80S complex, 80S ribosome, or for forming the entire 80S ribosome, you require the EIF5. Okay.
why this much initiation complex? They can directly say na, that this is binding, and everything is happening. Now they are forming again the initiation complex.
Because for yeah, elongation step, you will require this entire 80S complex. Okay.
Means after addition of everything, they are saying that this is the name of this particular thing, and then they is going to do this.
Yes, yes. There are different steps. So different steps will require hydrolysis of GTP. And then for binding to the particular site, energy will be required. So for these different steps, you require different initiation factors, and it has to take place sequentially, one by one.
Okay, so got it.
Now answer this question. So exchange of GDP with GTP attached to EIF2 is mediated by
IFB, 5B.
EIF5B. So here the question that has been asked is exchange of GDP with GTP.
Don't know, sir.
EIF, what was the function of EIF5B? EIF5B
It
was
was
helping in the
bringing
hydrolysis of GTP.
largest.
It was breaking down GTP to GDP.
Hmm.
EIF5B, not EIF2. EIF2 was bringing the initiator tRNA to the P site, right? But EIF5B, what was its function? That this EIF5B, sorry. So
Sir, it was bringing the largest ribosome, na, to the.
yeah, it was bringing the initiator tRNA. EIF2 was bringing the initiator tRNA. Yes, you are correct. EIF2
And
was bringing the initiator tRNA, and this EIF2 was attached to GTP, right? Now, for association of the 60S subunit, this GTP was getting hydrolyzed so that the 80S initiation complex can be formed. Okay, but now, yes, now this GTP has been hydrolyzed to GDP. Okay, now in the, for example, you have to do another cycle of translation. So you have to form another protein. Now this for now after this step, what happens? The EIFT, my EIFT is bound to EIF2 is bound to GDP. Now how this? So which protein is involved in the exchange of GDP to GTP? You understand the question?
GDP to
So
GDP.
exchange of GDP with GTP. So so
Sir, how how
initially
this
what was happening? You tell me. You tell me. EIF2 was there. EIF2 was bound to GDP. Right. Now once EIF2 is bound to GDP, it recognizes the initiator tRNA. This is my initiator tRNA. My initiator tRNA is attached to this methionine residue, and this initiator tRNA is recruited to the P site, by this EIF2 initiator protein, right? So my initiation factor 2 brought the initiator tRNA to the P site. Okay. So now when the 80S initiation complex has to be formed, this GTP got hydrolyzed to GDP. But again, if another protein has to be synthesized, then this GDP has to be again exchanged for GTP. So there is another protein which is involved in this.
then
Exchange of
EIF 2B maybe it would be the protein. 2B or 4A.
so answer is actually EIF2B only. Okay,
Okay, sir.
because this is a GDPS protein, EIF2B.
So how GDP can be converted to GTP? GTP can be converted to GDP, but GTP. This protein will bring the phosphate.
Uh, the, again, this GDP can be attached with another. So this GDP can be attached to another inorganic phosphate, and GDP can be formed, right?
Mm-hmm.
That is what is happening. So this is a GTPase protein. It has guanine nucleotide exchange factor. So this guanine nucleotide exchange factor can convert the GTP to again GTP. Okay. So the initiated tRNA is escorted to the small subunit by the 3 subunit GTP binding protein EIF2. EIF2 binds the initiated tRNA only in the GTP bound state. Okay. The complex between the so this is one important point you have to remember that this eukaryotic initiation factor 2, it can only bind to the initiated tRNA when it is bound to GTP. Okay, and this complex between the initiated tRNA and EIF2 is called the ternary complex. Okay. So this is a table which was there in your slides. Okay. EIF1, EIF1A, EIF2, EIF2B, EIF3. So different functions have been mentioned here.
I skip this table.
Hmm. So this is actually a summary of what are the different
This is
initiation
this
factors.
is this is important for net CSI or net or gate. This table.
PSIR net and the section B, they ask these type of questions. Which factor is involved in recognition of the 5 prime cap? Which initiation factor blocks the P site, blocks the A site and the E site?
Means this from this table it will come.
Yeah, from this table will come.
Okay, sir. The
So you
questions
see
are
here,
over.
this is actually a summary, EIF40. EIF40 binds to the 5 prime cap of mRNA. So remember a few initiation factors, EIF40. Then EIF2, remember that EIF2 remains bound to GTP and it is involved in recruiting the initiator tRNA. So few, then EIF, all
5B.
these EIF4, all these EIF4A, 4B, 4E, 4F, 4G, all these initiator proteins were what? They were all involved in they were all involved in bringing the mRNA to the 43S initiation complex, right? All these E4A, 4B here, if you remember the figure. So you see here
Yes.
all these 4A, 4G, 4B, they were all binding to the mRNA and bringing the mRNA to the 43S initiation complex. Okay. So it is not difficult. If you understand it, then then it will be easy to remember. And then there is this EIF3 where it was binding at the A site, right? First initiation factor binding to the 40S subunit and promotes further steps having GEF activity. So this is the guanine nucleotide exchange factor. Okay. EIF2B, EIF3. So having GEF activity, this is actually EIF2B. 2. Okay. Okay. So you see, this is the figure which where this entire translation initiation has been explained. So you see here, this is the 80S complex, which is composed of the large subunit 60S and 40S. So in the first step, you see what happened. The two subunits got dissociation, 60S and 40S, right? And then what are the initiation factors which are coming? EIF1A and EIF3. Then EIF1A and EIF3, these are binding to the, this is the A site and this is the E site. Okay, so these are binding to the A site and the E site, EIF1A and EIF3. And then this is your initiator tRNA. So initiator tRNA will remain attached to the methionine amino acid residue. Okay, and then this methionine residue, EIF2 GTP, EIF2 GTP, and this 40S ribosome complex, this is together forming the 43S initiation complex. And then there is this mRNA. This mRNA is being recruited by these EIF4 initiation factors, 4E, 4G, 4A, 4B, and then you get the
43S mRNA complex.
43S, and after this mRNA has joined, you get the 48S actually. Okay.
After
Some error.
mRNA joining and after the 60S subunit formation
Yes,
we will
yes.
get to.
Hmm.
Hmm.
You see here in the last step, what is happening? This GTP is getting hydrolyzed, and this GDP, EIF2 GTP is getting hydrolyzed to GDP, and again this GDP is again forming GTP. Okay, by attachment of another PI group. Now, hydrolysis of GTP bound to EEF2. Now, EEF2 is an elongation factor. Okay, not going into the details of elongation mechanism of eukaryotes. So in elongation, again, there are different factors. So hydrolysis of GTP bound to eukaryotic elongation factor 2 causes
Release of large subunit.
from
No,
the
no.
mRNA. Right? Ensures correct pairing of codon with anticodon, peptidyl transferase reaction, and translocation reaction. So in elongation, what has to happen? Elongation, the ribosome has to move on the mRNA, right? So actually answer is translocation reaction. Okay. So in translocation, what happens? So the ribosome moves to the next codon of the mRNA. So one by one, the ribosome is reading the codon in triplicates, right? So after one mRNA is the one codon is read, this ribosome has to move forward and read the next codon. So for moving for moving along the mRNA in the 5' to 3' direction, this elongation factor 2 is required. So always remember that 2 always will remain associated with GTP. Okay. For all the two that we saw today in prokaryotes, also initiation factor 2 was attached to GTP. In eukaryotic, initiation factor 2 was attached to GTP. And now in elongation factor, this elongation factor EF2 also remains to GTP, not to GTP. And when this GTP gets hydrolyzed, it supplies the energy to move mRNA. Okay, so actually what happens, the ribosome does not move, the mRNA moves. Okay, so if this is your ribosome and this is your, if this is your mRNA and this is your ribosome, this mRNA actually, uh, this ribosome remains stationary, but this mRNA moves so that the ribosome can read the further codons in this open reading frame. Okay. So the GTP gets hydrolyzed and supplies energy to move mRNA. EF2 and GTP complex recycles for translocation. About 6 amino acids per second are incorporated during the course of elongation of translation in eukaryotes. Okay, remember this point only, that EF2 and this GTP is required for the hydrolysis of GTP, which remains bound to EF2. It is required for translocation. Okay. So here also there are different elongation factors which are required. Now this is the last question, I think. During the process of translation, the addition of incorrect amino acid to the growing polypeptide chain can be prevented at dash step. So the correct answer is actually attachment of amino acid to tRNA. Okay, because at this step that the amino acid synthetase that was involved in attaching or the particular amino acid to the tRNA. So so that can be inhibited, or the addition of incorrect amino acid to the growing polypeptide chain. So depending on what is the anticodon, sequence. Based on that, a particular amino acid will be recruited, right? So, if you have to inhibit the incorporation of incorrect amino acid, you have to prevent the attachment of the incorrect amino acid to the growing polypeptide chain. So if you prevent the attachment of amino acid to the tRNA, then the incorrect amino acid will not be incorporated, right? So the answer is option A. Okay. This is dash acts as the GDP exchange factor for EFTU GDP. Now this is again in elongation. This protein is involved elongation factor TU. So this elongation factor TU GDP. This acts as the GDP exchange factor for EFTU. Okay, so after EFTU, GDP is released from the ribosome. A molecule of EFT bind to EFTU, causing the displacement of GDP. The elongation factor EFTs acts as the GDP exchange factor. So these act as the GDP exchange factors. When this EFTU GDP has to be replaced with EFTU GTP. These EFTs are actually required. So EFTs, you see here, these EFTs will only so that is what is explained, that the elongation factor EFTs acts as the GDP exchange factor for the EFTU. After EFTU, GDP is released from the ribosome. A molecule of EFTs bind to the EFTU, causing the displacement of GDP. So once this EFTs bind, this GDP molecule is displaced from this EFTU, and this GDP binds to the resulting EFTU EFTS complex, causing its dissociation. Now, when GDP will bind, this GDP will cause what? GDP will cause the it will cause the dissociation of this
ribosomes.
EFTU EFTS complex into free EFTs and EFTU GTP. Okay, so EFTU is actually required. So for the recruitment of the initiator tRNA, the what was required? EIF2, right? EIF2. Now for the recruitment of subsequent tRNAs, now the subsequent tRNAs will be recruited at the A site. Okay, so for the recruitment of the subsequent tRNAs, this EFTU is required. Okay, so this EFTU will. So this EFTU, when it is bound to GDP, it cannot. This GDP has to be replaced by has to be displaced so that this EFTS can bind. Now, once this EFTU complex is bound to GDP, this EFTs are displaced, and now this EFTU can recruit the next tRNA. So this is the subsequent tRNAs which will be recruited after in the elongation step. Okay. Now there is one question related to post-translational modification. So this is so in post-translational modification, the topics that were discussed was phosphorylation, ubiquitinylation, right? So related to ubiquitin, this question, ubiquitin is attached to E1 enzyme via dash linkage. So the proteasomal ubiquitin pathway that takes place in cells that you have to understand. So there are different enzymes E1, E2, E3. So which enzyme is involved in what? So that pathway. I'm not going into too much detail. So basically, this answer is actually thioester bond. So what happens is that when a protein has to be degraded, so you know that when protein has to be degraded, a ubiquitin moiety is attached to it, which marks the protein for degradation. Okay, so how this ubiquitin group, how a ubiquitin moiety is attached to the protein. So for this ubiquitin attachment of the ubiquitin, there is this ubiquitin pathway where there are three main enzymes involved, E1, E2, and E3. So first step, what happens is the carboxyl end of ubiquitin. So this is the carboxyl end of ubiquitin. So needs to be activated for protein ubiquitination. So how is this activated? This activation is accomplished when a protein called a ubiquitin activating enzyme, E1. E1 uses ATP hydrolysis energy to attach. So this is the ubiquitin activating enzyme E1. This uses ATP hydrolysis to attach a ubiquitin to itself through a high energy covalent bond. So this ubiquitin group is attached to the E1 enzyme through this thioester bond. This S bond you are seeing, this is the thioester bond, and then this E1 passes the activated ubiquitin to one of a set of ubiquitin conjugating enzymes E2. So now this is then transferred to E2, each of which acts in conjugation with a set of accessory E3 proteins called ubiquitin ligase. Okay. So once you have this ubiquitin, then this ubiquitin group can be transferred to the target protein. So you have a target protein which has, for example, this is a misfolded protein. You have to degrade the misfolded protein, then this ubiquitin group has to be transferred or transferred to this misfolded protein. Then so for what will be required, this moiety is then transferred to E3 ubiquitin ligase, which will transfer this ubiquitin group to the target protein. Okay, so this is ubiquitin proteasome degradation pathway. And in this pathway, you can get this mechanism how it is done. So remember that in the first step, ubiquitin is attached via the thioester linkage. So this is the one. So this is the last question for today. So um, I hope some things were clear. I will take care that from next class. I will add the entire concept so that you can better understand all the concepts. Okay.
Sir, or you can add video also.
Okay. Okay. Yeah. Video also I can add in the short videos. I can attach
Yes.
to the slide. That things will can visualize what is happening. Okay.